Our topic for today is T cell development. Uh, we had already discussed the different cells and the organs of the immune system. So with that knowledge in the background, this is a very preliminary picture showing you the sites of hematopoiesis. Hematopoiesis means development of the immune cells from a single hematopoietic stem cell. So you can see the site of hematopoiesis in the fetus is the yolk sac, the liver and the spleen, as you can see it here. As we mature, as we tend to grow up, the site of hematopoiesis gradually shifts into the axial skeleton and the distal long bone. So you can see that the sites of hematopoiesis are the distal long bones. So we have these bone marrows later on in our life where this site of hematopoiesis is observed, even though in the fetus, the initial site of hematopoiesis is liver and spleen. This is a recapitulation of the progeny of the T and the B lymphocyte since we are doing the T cell development. So just to recapitulate, the T cell origin is in the bone marrow and the precursor cells move into the thymus. Within this thymus, the T cells mature, develop, and ultimately they come out from the thymus and then migrate to different peripheral lymphoid organs. This is also recapitulation. This is showing you the structure of the different primary. There are basically two categories of primary lymphoid organ, the thymus and the bone marrow. So you can see the location of the thymus. And when we talk about the thymus, it is basically a bilobed organ located, as you can see the location of the thymus. It is located in the thoracic cavity overlying the heart and the major blood vessels. And each lobe is organized into separate form. Each lobe is organized into lobules, which is separated from each other by connective tissue. And this is a brief structure of the thymus. We had discussed thymus in details when we did the lymphoid tissue. So just a recapitulation. Within each lobule, the lymphoid cells are arranged into thymocytes. So you can see the thymocytes. Now the thymus this is a picture of a capsule, and you can see that each capsule can be divided into the cortical region and the medullary region. And two important cells are found both in the cortical and the medullary region are the epithelial cells that play a big role in schooling of the T lymphocytes. The outer part, if you look into the cortical area, the outer part is packed with relatively immature proliferating thymocytes. The inner medulla contains the more mature cells. So it is evident that as the lymphocytes moves through the cortex into the medulla, the maturity of they gradually get themselves transferred from an immature thymocyte into a mature thymocyte. This is another beautiful picture of the thymus. In fact, we discussed this picture earlier when we studied the thymus. This picture specifically shows the three categories of important cells present in the thymus. Again, you can see the thymus has been divided into the cortex and the medulla. And the three basic category of cells that we can observe here, the epithelial nerve cells. As you can see the function, they sustain the proliferation of the progenitor T cells, the T cells, the precursor T cells, the mother T cells, which are coming from the bone marrow into the thymus. So these epithelial nerve cells in the outer cortex sustains the proliferation of this progenitor T cells through the cytokine production, and the cytokine mainly produced in this case is interleukin 7. Then if you look into another category of cells, the cortical thymic epithelial cells, these cells are responsible for one form of schooling of the T lymphocytes, which is actually known as the positive selection of maturing thymocytes. In the positive selection, as we'll do it in detail later on, the T lymphocytes which are showing affinity for either class 1 or class 2 MHC molecules are selected. Remember, when a T lymphocyte is born and the progenitor enters the thymus, initially the T lymphocyte for some time expresses both the CD4 and the CD8 receptor. I hope you remember the importance of CD4 and CD8 receptor. Now, since both the CD4 and CD8 receptor are present on a single lymphocyte, a time comes when the lymphocyte is told you keep either one of the receptors. Because a lymphocyte either has to be a CD4 positive lymphocyte or it has to be a CD8 positive lymphocyte. It cannot be both. So this is positive selection. And this to some extent is done by the cortical thymic epithelial cells. And when you look into the medullary region, there are also thymic epithelial cells present in the medullary region. They display a large variety of organ-specific self-peptides because the T cells also need to realize which are self-peptides and which are non-self-peptides. If there are too much reactive T lymphocytes towards a self-peptide, then these lymphocytes, by a way of negative selection, are forced to die. So as you can understand, through this cortex and via these two category of cells, two very important schooling of the lymphocytes is done. Another important cell that is mostly observed in the medullary region are the Hassel's corpuscle in the thymic medulla. Till date, their function is unknown, but it has been observed that they appear to contain degenerating epithelial cells rich in high molecular weight cytokeratins. So this is basically they have been seen that they contain this, but exact function in the development of T cells have not yet been discovered. This is a rough summary of how the T cells moving through the thymus develop. Since you have studied T cell signaling, you have studied T cell receptor. So when we are starting this topic, I have this basic concept that you are aware of the T cell receptors, the signaling processes, as well as the MHC presentation. So whenever a T cell, precursor T cell enters into the thymus, the first thing it gradually develops are the T cell receptors, that is the alpha beta receptor. And there is also this gamma delta population. After this receptor development, that is alpha beta receptor, then comes a stage which is known as positive selection. In positive selection, the T cell interacts with MHC class 1 or MHC class 2 presented by the thymic epithelial cells present there. The thymic epithelial cells at the cortical region acts as the antigen presenting cells in this case. And those lymphocytes which interact via MHC class 2 become CD4 positive, and those T lymphocytes which interact via CD8 or CD8 co-receptor and MHC class 1 become CD8 positive. So there is this event of positive selection. Once these lymphocytes have been positively selected, then comes an event of negative selection. The negative selection takes place to ensure that T lymphocytes do not react vehemently with any self-peptide being displaced. So all those T lymphocytes that interact with very high affinity for a self-peptide. Self-peptide means a peptide of our own body. For a self-peptide, they are not allowed to leave. They are forced to die. So this is negative selection. And finally, once this positive and negative selection has occurred, then only the lymphocytes moves out of the thymus. These are mature lymphocytes, and then they travel to different lymphoid organs. So as you can see, in the initial stage of the development, the precursor progenitor T lymphocytes moves from the bone marrow into the thymus, and in the thymus, via their schooling, via the development of their receptors, Via positive and negative selection, when mature thymocytes have been formed, these lymphocytes then move to the various lymphoid organs present in the body to continue their function for which they are destined. So, if you look into this picture, let me tell you the abbreviation of two uh, things. One is the DN. Keep it in mind, DN means double negative cells. And I'm saying DN, double negative. And why double negative? Whenever we are talking of T lymphocytes, as you have understood by now, the CD4 co-receptor or the CD8 co-receptor is very important. The total lymphocyte progeny is distinguished into CD4 positive cells or CD8 positive cells, and they form two different functional aspects of the adaptive immune system. When I'm using the word, or when the picture is using the word double negative, it means that these two co-receptors have not yet developed in the lymphocyte. When I'm using the word double positive, that means the lymphocyte has a single lymphocyte has both the CD4 positive CD4 receptor and the CD8 receptor. When I'll be using the word single positive, that means the lymphocyte has either the CD4 receptor or the CD8 receptor. Now let us look at this picture. I have given a supporting picture, but when I am talking about the development of T cells within the thymus, restrict your view to this picture. As you can see from the picture, the T cell precursors from the bone marrow have traveled to the thymus via the bloodstream, and there they ultimately develop, as you can see at the end, into mature thymocytes. Now, during this development, each stage of development occurs in a specific microenvironment and is characterized by specific intracellular events. So you see the hematopoietic stem cells coming through the blood, and the hematopoietic stem cells or the T cell precursor enter the thymus. Once this immature thymocytes have entered the cortical region, these thymocytes, as you can see, are double negative. That means they do not have the CD4 or the CD8 co-receptor. These double negative cells then passes through 4 stages: double negative 1 to double negative 4. And during this passage from double negative 1 cell to double negative 4 cell, as you can see, the T cell receptor development takes place. If you remember the T cell receptor, we had this alpha chain, beta chain. Some T cells had the gamma chain and the delta chain. So from the transition between double negative 1 to double negative 2, you can see that the TCR beta locus rearrangement occurring. The beta chain has developed, and the T cell is gradually being committed. During this transition from double negative 2 to double negative 3, some T cells develop the gamma delta chain and they become the gamma delta T cell. Once these T cells become the gamma delta T cells, they do not enter the pathway of the alpha beta differentiation. As you can see, they migrate out because the alpha gamma delta T cells do not have the CD4 or the CD8 co-receptor. So there we see a branching out of the gamma delta T cells. Now as the cell proceeds to double negative 3 stage, beta chain selected, double negative 4 chain, double negative 4 stage alpha chain development occurs. So it started with double negative 1, and when it comes to double negative 4, we see that the alpha and the beta chain, that is the T cell receptor development, has been completed. And the TCR gamma delta chain, that category of lymphocytes, has made an offshoot from the main stream and has separated out. Now when we have this double negative 4 stage and we have the T cell receptor alpha beta chain completely developed, the cell becomes transiently double positive. That is, both the genes for CD4 and CD8 are expressed, and both the co-receptors, the T cell lymphocyte is now expressing on its surface both the CD4 co-receptor and both the CD8 co-receptor. So the cell has become transiently double positive. I'm saying transiently because this is not a permanent phenomenon. After the cell has become transiently double positive, you can see there is this phenomenon of positive selection. I'll come to that separately. So when we are having this event of positive and negative selection one after another, we finally get a population of single positive, either CD8 positive cells or CD4 positive cells. So initially there is positive selection. The positive selection ensures that the lymphocyte will be either CD4 positive or CD8 positive. All double positive cells die off. All cells who fail to express only one co-receptor, those who still are having both the co-receptors die off. So there is this single positive cells now. This selection is known as positive selection, and these single positive cells are then again tested. They go through another round of examination where the single positive cells are seen whether they react heavily with self-peptides. If so, then those cells are also allowed to die. Only moderately reactive T lymphocytes with self-peptides are selected. This is negative selection, and after negative selection, we are getting mature CD8 positive and CD4 positive T lymphocytes, and this mature CD8 positive and CD4 positive T lymphocytes now migrate to the peripheral tissue. So that is in nutshell the development of the T lymphocytes through the thymus. Now let us come to these two important steps of selection, the positive selection and the negative selection. So if we look into the two basic categories of selection, the positive selection takes place in the cortex and the negative selection takes place in the medulla. The positive selection is a simpler one, as we just now saw that the double negative cells DN4 have both the CD4 and the CD8 co-receptors, and these double positive thymocytes now express the TCR alpha beta dimers. So they have the T cell receptor also. Now they start interacting with the cortical thymic epithelial cells. The cortical thymic epithelial cells acts as an antigen presenting cell. These thymic epithelial cells can express both MHC class 1 molecules and MHC class 2 molecules. So in a way, they can express foreign peptides complexed with MHC class 1 molecule and foreign peptides complexed with MHC class 2 molecule. Now, since this particular double positive cell is a cell which is having both the CD4 and the CD8 co-receptor, when a lymphocyte interacts with this thymus epithelial cortical cells, it will either recognize the MHC class 2 molecule via CD4 or MHC class 1 molecule via CD8. If it recognizes the MHC class 2 molecule via CD4, then its CD4 receptor is selected, CD8 is automatically down-regulated. Vice versa, if this is a random selection, if a double positive lymphocyte interacts via CD8 with MHC class 1, this is the first interaction, then its CD4 is down-regulated and its CD8 is up-regulated. That is, it becomes CD8 positive cell. So the question is that the double positive lymphocyte interacts with which receptor? If it interacts with CD4 co-receptor, then the CD4 is selected the other one is switched off. If it interacts with the CD8 receptor, then the CD8 is selected and the other one is switched off. In this way, via interaction with the thymic epithelial cells, as you can see, and through MHC class 1 and class 2 receptors, some lymphocytes become CD4 positive cells and some lymphocytes become CD8 positive cells. Some die by neglect where there is no interaction, and you can see the percentage of cells dying by neglect. So the total amount of lymphocyte precursors that are coming is only 2 to 5% that properly interact with the thymic epithelial cells and get selected. 2 to 5% have high affinity interaction, they are forced to die off. And as you can see, if there is no interaction, they are also forced to die off. So once these cells are the CD4 positive, so we have this positive selection. So the double positive cell has now become either CD4 positive cell or CD8 positive cell. Once this selection has been completed, the T cell then undergoes a second set of selection, that is the negative selection. As you can see, the single positive cells have now migrated to the medulla, where they are exposed to the medullary thymic epithelial cells. In the medullary thymic epithelial cells express tissue-specific, our own tissue-specific antigens. And if the T cells are seen overreactive to this tissue-specific antigens, the single positive T cells are seen overreactive to this overreactive to the self antigens, then these single positive cells are also forced to die off. Only those single positive T cells which are tolerant to our self antigens then mature to give rise to mature CD4 positive and CD8 positive T lymphocytes. So, thus now, after this phenomena of positive and negative selection taking place in the cortex and the medulla respectively, the T cells have now matured. This is another picture showing you this is a beautiful picture. This is showing you, look at the different, I said that every stage of the thymocyte development through the cortex requires a host of factors, and the microenvironment changes as the double negative cell travels towards the double positive cells. You can see the difference in this is the signaling that starts in the double negative 2 cells. You can identify the TCR alpha beta, the LCK, the ZAP70. So that is the signaling that takes place during this time, which I just explained. And these are the different types of receptors that express when the double negative 1 cells part travels ultimately to become the double positive cells. And this is a simple death by neglect. Death by neglect occurs when there is no antigenic exposure. So there's no exposure. So the T cell receptors, there's insufficient signaling. We have understood the signaling cascade in our earlier class. So if there is insufficient signaling, automatically the T cells will not be activated properly. And if the T cells are not activated properly, they are allowed to die. So this is a phenomena which is death by neglect. As I discussed the positive selection, you can see that when these double positive cells are interacting with the CD4 positive co-receptor with the epithelial cortical cells, then they become CD4 positive. If they are interacting with the CD8 receptor with the epithelial cortical cells, they become CD8 positive. And this is again negative selection. If the single positive cells in the medulla interact very strongly with the medullary epithelial cells for self-peptides, then these cells, single positive cells, are also forced to die by negative selection. Further, negative selection also occurs in the cortex. Remember that double positive cells at times may interact heavily with the self-peptides, at times displaced in the cortical region. then those cells are forced to die. I'll just show you once more. See, there's a phenomenon of negative selection also occurring in the cortical region. So in the cortex also at times cell peptides may be displaced, and if these double positive cells are seen to interact heavily with these cell peptides, then negative selection also occurs in the cortical region. Here the negative selection is occurring for double positive cells. In the medullary region, the negative selection is occurring for the single positive cells. Now, there are three models which have come up which proposes that how this lineage commitment is taking place. When I talk about lineage commitment, what does this mean? What does this word lineage commitment means? You see that we started with a thymocyte, precursor thymocyte, and you also saw that after passing through double negative 1 to 4 stage, the precursor thymocyte became double positive. So it had both its co-receptors. Now the question is that how these both co-receptors from this both from a double positive cell having both the co-receptors, how the cell became single positive. So that is commitment of lineage. So the double positive cell is now committing itself to either the CD8 positive cell, or the double positive cell is committing itself to a CD4 positive lymphocyte. Remember, since you have studied the cells of the immune system earlier, this lineage commitment is very, very important. Whenever a double positive cell becomes CD8 positive cell, a CD4 receptor is downregulated. Not only that, it becomes a cytotoxic killer lymphocyte. They become the killer arm of the adaptive immune system. Similarly, whenever a double positive cell becomes a CD4 positive, CD4 positive single positive T lymphocytes, its CD8 co-receptor is downregulated and switched off, and this CD4 positive cell forms a pivotal link between the B lymphocyte and the CD8 positive T lymphocyte. So the moment their one receptor is being switched off, they are losing one functional facet and they are being committed for a very particular function. So that is lineage commitment. Now there are three models to explain this lineage commitment. The first model is the instructive model. In the instructive model, as you can see, their interaction with their dendritic cells or the epithelial cells present in the thymus is the one that is instructing this double positive cell to decide their fate. So if the cell, this is just a repetition, if the cell is interacting via CD8 co-receptor, then the CD8 engagement signal starts and the cell finally becomes a CD8 positive cell. So we have a low CD4 receptor and then gradually you can see it's becoming CD8 high and finally the cell is becoming CD8 positive cell. Similarly, if the double positive cell interacts via the CD4 co-receptor, then accordingly CD4 receptor, the gene expression for CD4 is becoming high, the CD4 receptor expression is becoming high, and it is becoming a CD4 positive cell. So this is the instructive model. Now, according to the stochastic model, the downregulation of the either or receptor is random. So if the lymphocyte is able to bind to antigen and MHC class 1, the double positive binds to antigen and MHC class 1, CD8 positive T cells. If they are not able to bind to MHC class 1, then we see death of some of the lymphocytes. Again, random selection of CD4. So you can see this is random selection. So randomly there is a high expression of CD8, interaction with MHC class 1, CD8 positive cell. Those cells with a high expression of CD8 but failing to interact with MHC class 1 die. Again, random selection of CD4 positive receptor. So CD4 high expression. If they are able to interact with MHC class 2, CD4 positive cells. if these cells are unable to interact with MHC class 2 apoptosis. So if we have a population of around say 100 lymphocytes, randomly 50 have high CD8, 50 have high CD4. That's a random selection as per this model, as per the stochastic model. And this initially high CD4 or CD8 expressed lymphocytes then interact accordingly with class 1 and class 2, and further stabilization of the CRO receptor is occurring. And finally coming to the kinetic model. Now, the kinetic model tells us the story that all lymphocytes, this is a double positive cell, and the T lymphocyte is now engaging with the thymic epithelial cells, and all receptors at the beginning, all the lymphocytes, all the thymocytes at the beginning are having a higher expression of CD4 positive co-receptor. So the CD8 co-receptor is very low. And the CD4 positive, where is this? Single positive cells starts developing. Now what happens? When the CD4 positive cell starts developing initially and they increase in number, the number has increased. So when they have increased in number and during that process, CD8 receptor expression has been diminished. So there are two things happening. CD4 positive cells number has increased, and the CD8 receptor expression till now has been diminished. A time comes when this down regulation of the CD8 receptor interrupts or intercepts, I'll not say interrupts, intercepts a TCR CD8 MAC class 1 signal. Suddenly, one of the CD8 intercepts a MAC class 1 signal, and all of a sudden, this disrupted TCR signal, that lymphocyte which has experienced a sudden CD8 receptor signal, that lymphocytes then starts proliferating, and the cells in which the CD8 interruption has taken place, in those cells, the CD4 automatically gets down regulated, and these cells then starts proliferating. So this is a kinetic signaling model actually tells us that it is the signaling decision that helps in converting a double positive cell to a single positive cell. As per this kinetic model, keep it in mind, initially all the cells have this CD4 positive receptor signaling. Their CD8 expression is low but not switched off. All of a sudden, if one of the lymphocyte can receive a signal through the CD8 co-receptor, that then automatically switches on a CD8 co-receptor. The CD4 in that cell becomes down-regulated. Simultaneously, some of the cells which are specifically having their signals via the CD4 co-receptor are selected as the CD4 positive single positive cells. So these are the three models for lymphocyte selection. For the selection of a single positive cell from a double positive cell. And now moving on to another point. Just a second. So whenever now we have these mature CD4 positive cells and mature CD8 positive cells, there is another form of the development that takes place in broader terms that takes place is that the T cells can be further divided depending on what cytokine the environment is secreting. So we have this mature CD4 positive T cells. They are naive. Naive means the CD4 positive mature T cells have not yet faced a foreign challenge. During its schooling, it had faced a foreign challenge, but after it has been schooled, after it has come out from the thymus, it has not yet faced a foreign challenge. Now, whenever this naive CD4 positive T cell interacts with an antigen present T cell, Then depending on what kind of cytokines are being liberated in the microenvironment, a single CD4 positive T cell can polarize itself into, as you can see, 7 varieties of CD4 positive cells. So we can have the T regulatory cells from, see, this is the differentiation of a single CD4 positive T cells into 7 category of cells based on the cytokines that are liberated in the microenvironment when it is interacting with an antigen presenting cell which is displaying a foreign peptide. So the different polarized condition a CD4 positive T cell can achieve are T regulatory cells, T helper 17 cells, T helper 2 cells, T helper 9 cells, T follicular helper cells, T helper 1 cell, and T helper 2 cell. It is this category of the helper T cell that can interact with the CD8 positive single positive cells. The other category of T cells are not able to interact with the CD8 positive T lymphocytes. Just to tell you here, whenever we are having this basic differentiation of CD8 positive helper cells and CD8 positive T lymphocytes, the CD8 positive T lymphocytes are known as cytotoxic T lymphocytes because they kill their target cells. So they are toxic to the cytoplasm of the target cells, hence cytotoxic T cells. So this category, this helper 1 category cells are the only CD4 positive T cells which can interact with the T cytotoxic cells. The others, they have defined function. For example, if you look at the polarizing cytokines. In presence of interleukin 2 and TGF-beta, you can see that all these cytokines, look at the colors, they are all expressing different transcriptional regulators. So when these lymphocytes are receiving, when a CD4 positive lymphocyte is receiving a IL-6 cytokine, IL-23 cytokine or TGF-beta cytokine, you see upregulation of ROR gamma T. That transcription regulator is taking place and the naive CD4 positive T lymphocyte is becoming a T helper 17 cell. So this is how the polarization of T lymphocyte, CD4 positive helper T lymphocyte takes place, and we get, as you can see, 7 varieties of effector T lymphocyte, T helper lymphocyte, 7 varieties of effector CD4 positive lymphocytes from a single CD4 positive helper cell. And as you can see, their role is also very different. If you look at, let me remove this ink. If you look into the T regulatory cell inhibits anti-tumor response. T helper 17 cells, autoimmunity. T helper 2, allergy. T helper 9, autoimmunity. And you can see each is having its distinct cytokine set. T follicular cells helps in B lymphocyte maturation. T helper 1, tissue inflammation, helps to overcome tissue inflammation, combats intercellular pathogens. And T helper 22, you can see inflammatory skin disease. So this is the polarizing signals that the CD4 positive T helpers, once if the polarizing signals are received, a single CD4 positive T helper cells can be converted into this polarized variety of the T helper cells. And the 2 basic polarizing signal, I showed you the 7 categories of polarizing signal, but the 2 basic polarizing signal is the T helper 1 and the T helper 2. These 2 are very famous. T helper 0 means the naive CD4 positive cell. Depending on what kind of infection we are having, the 2 basic, the initial common CD4 positive helper cell can differentiate into the helper 1 category or the helper 2 category or the helper 17 category. As you can see, for intracellular infections, we are having the helper 1 category. That means any cell of our body which is infected with a virus, this is an intracellular infection. So this is infected with a virus. Now these viral proteins are displaced via, if you remember, MHC class 1. So the immune system needs to kill this cell. Now how to kill a cell of your own body? The way to kill a cell of your own body is to send a lymphocyte with a message that send the signal of extrinsic death pathway to that cell. And that is possible by the T cytotoxic cells. You have studied apoptosis. If you remember, you have studied the Fas ligand. All the T cytotoxic cells are able to express the Fas ligand. So whenever there is this intracellular infection, T cytotoxic cells, CD8 positive T cytotoxic cells are activated. They express their CD Fas ligand and they move towards this target cell. On the surface of the target cell, the Fas receptor trimerization occurs and the message for extrinsic death pathway is switched on and the cell is allowed to die apoptotic death. There's another way of killing the cell also. I'm not getting into that detail today. So this is one important interaction between the helper 1 category and the T cytotoxic cell. So as you can see, this is just extra information. So that was about the T cell development and the T cell polarization. But when I was doing this signaling aspect of T lymphocytes with you all, again and again, I told you that there are 3 signals we needed for a proper activation of T cell lymphocyte. The first signal was the signal through the T cell receptor itself. But this signal was not enough. If you remember, there was another co-stimulatory signal that took place between the B7 CD28 and CD8086 molecule. Once this signal 1 and signal 2 was achieved, if you remember, the signaling cascade started and early genes were expressed. When the early genes were expressed, the lymphocyte is capable of producing its cytokines. The lymphocyte is capable of expressing its own, the receptors for the cytokines. So there is an autocrine loop, and this is the third activation message. So whenever there is this T cell activation, there is this signal 1 and signal 2 is mandatory, and hence this T cell activation process is also known as 2-signal hypothesis. This is a very simplified picture of showing you what happens actually. You see a naive T helper cell binds with the MHC peptide complex helper cell, so it is binding with MHC class 2. Next, what you can see, this is signal 1. Next, you can see signal 2, that is interaction of CD28 with B7. Then you can see the signal 3, cytokine help, and you can see the T helper effective cells. One thing, so we are having a helper effect, so the T helper cells have become active. But in the process, what they did is that initially the antigen presenting cells which were interacting with the helper cells were naive. Even they were learning while interacting with the T lymphocytes, and in fact their signaling and the signaling process operative in this antigen presenting cells was activated. So once a T helper cell has been activated and this antigen presenting cell and the helper cells free off from each other, in the process this dendritic cells have also become activated. So these are licensed dendritic cells. These licensed dendritic cells now can interact with naive T cytotoxic cells via MHC class 1, and in the process can activate the cytotoxic T lymphocytes. So keep it in mind that whenever there is an immune reaction occurring in the body, the first arm of the adaptive immunity is the T helper cells. T cytotoxic cells are not activated automatically. The T helper cells react with the antigen presenting cells, in turn activating the signals of the antigen presenting cells. as well as activating the signals within themselves. These activated antigen presenting cells or the licensed antigen presenting cells then go and activate the T cytotoxic cells. So this is whenever a cytotoxic T lymphocyte is activated, there are different ways of killing a target cell. I just now told you that one of the famous way of killing a target cell is via the death receptor pathway. There are some other molecules also, I'm not getting into the details. So that was basics about how T cells develop in the thymus, how they move through the thymus, how they get selected to a single positive population via positive and negative selection, the models operative in the process of positive selection, and how the CD4 positive helper cells then can differentiate into 7 categories of polarized cells based on the cytokine environment. And we also just understood that there are 2 signals followed by the third signal that is very much essential for the activation of a helper T cell first, and only when an helper T cell is activated, then can a T cytotoxic cell can be activated. So with this, we end our class of T lymphocyte.